So, starting in chapter 7, we are going to start talking about neuroplasticity. So, we're going to take a couple different takes on it. We're going to talk about neuroplasticity and learning and how changes are made in the nervous system, and then what happens after injury, which is sort of where we get into it on the rehabilitation side of it. So, the first part of this lecture will just be hopefully short. About neuroplasticity, and then we will talk about what are the effects on healing after an injury. And by injury, I mean anything, onset of disease, a cerebrovascular accident, brain injury, anything. Okay, so our learning objectives for this section. I want you to be able to describe the changes in regions of brain activity from when you're initially learning a task to when you have mastered the task. And there's a difference. I want you to be able to define habituation and give a practical example of it. How fast is the recovery growth of damaged axons in the peripheral nervous system? And I want you to be able to list 3 reasons why central nervous system lesions are generally thought to be irreversible. And why can functional recovery happen after a central nervous system lesion, even though they are irreversible? So you can have recovery of function, even though those lesions are not going to heal. I want you to be able to list 3 chemical end products of an ischemic attack that the end products result in cell death that results from excitotoxicity. Well, what is excitotoxicity and what causes it? And what is the optimal amount of time, according to research, between stroke injury and the initiation of rehabilitation? It's sooner than you think. And how can the intensity of rehabilitation after stroke influence the recovery of neuronal function? All important things to think about in rehabilitation. So neuroplasticity is the ability of neurons to change their function, their chemical profile, or their structure. So by their chemical profile, we mean the amount and types of neurotransmitters produced. And more than that, the amount and types of receptor channel proteins that are produced. Because remember, the DNA in the soma is coding for proteins. It's coding for neurotransmitter proteins. It's coding for structural proteins. It's coding for receptor membrane proteins. And so what makes the neuron change its function, change the amounts and types of neurotransmitters it's producing? So neuroplasticity as a term is used to encompass the following mechanisms. Habituation is one of them, and we use that a lot in physical therapy. Experience-dependent plasticity, which is the fancy neuroscience way of saying learning and memory. So how does our brain change in response to what we experience? And then cellular recovery after injury. So also a very important idea in physical therapy. So habituation, by definition, is a decrease in response, and we mean neural response, to a repeated benign stimulus. So habituation is one of the simplest forms of neuroplasticity. And really, the cellular mechanisms that are responsible for habituation are not completely understood. So we know that it happens, that nerves accommodate and habituate, but we don't really know how. So last quarter in modalities class, when we're doing electrical stim, we talked about using modulation to prevent accommodation and prevent fatigue. We also mentioned that nerves accommodate faster than muscles, and muscles fatigue faster than nerves. So nerves have that ability to accommodate. So the idea of neuroplasticity and habituation has been around for a long time. In the 1800s, a researcher named Charles Sherrington conducted a study on reflexive behavior and habituation. He was doing it with rats because a lot of basic neuroscience is done with rats because they have a similar chemical profile to human beings, interestingly enough. So Charles Sherrington did he did a mildly painful stimulus to a paw So probably used a monofilament to stimulate the paw. The animal would withdraw its paw. After doing that same benign, non-harming stimulus for a long time, the animal finally just stopped withdrawing its paw. It just got habituated and it wasn't irritating or painful anymore. A simple example of habituation is you get a new watch for Christmas and you start wearing that watch and you can feel it on your wrist. You feel that new sensory information. And you notice it all day long. Well, eventually that repeated benign stimulus, you get used to it, you habituate to it, and then you can't feel it at all. And then actually sometimes when you take your watch off, you feel like, oh, there's something on my arm, but really you're feeling the absence of that repeated benign stimulus that you have habituated to. So physical therapy and occupational therapy are often used techniques and exercises intended to decrease the neural response to a stimulus. So sometimes we're doing habituation we're trying to diminish the sensory response to an injured area. After an injury, the nerves in the area are hypersensitive. We might be doing light touch or using something like a washcloth or a rough surface to stimulate the area to decrease the neural response, because we're trying to get the person used to being touched, or just the sensation of clothes touching your body sometimes is irritating in a newly injured area because of that heightened neural response, and we're trying to decrease response to the stimulus. So experience-dependent plasticity is another way of saying learning. So we have these experiences, they change our nervous system and become semi-permanent, hopefully some more permanent than others. But learning and memory is a complex process which involves persistent long-term changes in the strength of synapses with the repetition of a task. So I always say to people when we're exercising in physical therapy, well, when you're learning a tennis serve, you don't just practice it once and then say, yep, I got that. You practice it over and over and over again until you know it. And that new experience-dependent plasticity requires growth of new synaptic connections. They're actually, they can see with imaging the growth of dendritic spines when people learn a new task, which is pretty cool. So you're building a better nervous system. You're building a nervous system that is responsive to what you're trying to do with it. So we have a lot of potential synapses in our nervous system that aren't being used. And when we do something over and over again, the neuro world way of saying it is neurons that fire together, wire together. So the more you do something, the more you build those connections and you unmask silent synapses that were just waiting there for you. And maybe ones that you're not using get pushed by the wayside. But during the initial phases of motor learning, this is motor learning in particular, so you're learning that tennis serve, large and diffuse regions of the brain are active. So when they do functional MRIs now, in the learning of a new task, they see all different areas in the brain that are active, that are lit up on the functional MRI. When the tasks are repeated, so now we're doing that serve over and over again, learning our form, the number of active regions in the brain are reduced. When we're finally really good at it, and you could do that perfect serve in your sleep, when the motor task is learned, only small distinct regions of the brain show an increased activity when performing the task. So the other analogy that I like to use is when you're first learning how to drive, you're using a lot of your brain, you're gripped onto the steering wheel, your hands are at 10 and 2 or whatever they say now, I don't know. and you really can't focus on anything else, and somebody else is in the car and you say, oh, don't talk to me, I'm trying to focus on driving. After you've done it a while, now you're driving down the street, checking your email and drinking coffee. Hopefully not to that extent, but we're not using as much of our brain after we have mastered the task. So initially, large and diffuse regions of the brain are active. The number of active regions slowly reduces as you repeat the task, and then when you've got it mastered, it's only small distinct regions of the brain that show increased activity when performing the task. Pretty neat, right? So you can actually see changes in the brain. So that type of learning requires synthesis and activation of new proteins with a repeated stimulus. The synthesis and activation of new proteins changes the neuron's excitability, and it promotes the growth of new synapse connections, particularly in dendritic spines. So those are the little projections from the dendrites. So it actually requires growth of new synaptic connections. There are two different types of changes that are common in this experience-dependent plasticity, and they're called long-term potentiation and long-term depression. So long-term potentiation converts silent synapses to active synapses. This is the cellular level of learning. You know how I've been sort of pounding on it since the beginning of the program, that you have to give yourself time to encode that information. We have to have time for that cellular level of learning. When we're creating that, basically we're changing the postsynaptic membrane, we're growing new dendrites, we're activating new synapses, and that's what is resulting in our learning. That's how we're acquiring new knowledge. And it's also involved in neural recovery following an injury and when you're trying to improve your memory. So the two main types of plasticity in learning and memory formation are long-term potentiation, where you are creating more excitatory glutamate synapses. Those are those ones in the central nervous system, the excitatory ones. Long-term depression is where you're decreasing the number. So different forms of long-term potentiation and long-term depression can occur simultaneously depending on the type and location of the synapse. So when I was saying you're reinforcing these new synapses, you're strengthening existing synapses and you're creating new ones, and the ones you're not using sort of go by the wayside. So the ones you're not using, that's the long-term depression. The ones you're using more, that's the long-term potentiation. So the idea is that you can make change by what you're doing. So you're learning new tasks, you're learning new ideas, you're learning new information, and that is changing your nervous system. Pretty cool, right? So the glial cells are involved in this experience-dependent plasticity in learning. So the communication between astrocytes and neurons occurs via the release of neurotransmitter by the neuron that stimulates the release of gliotransmitters by the astrocyte. So they don't really know the exact role that astrocytes have, but those gliotransmitters, which are the transmitter molecules that astrocytes create, modulate neuronal activity and synaptic transmission. So they sort of act like neuromodulators in the area. It's thought, although not proven how, but it's thought that astrocytes influence synaptic plasticity through modulating that neurotransmitter release and receptor expression at the postsynaptic membrane. So there are studies that are done with rats where they're raised in what they call an enriched environment, where they have problems to solve and things to do, and it's like rat summer camp. And they show increased contact between astrocytes and neurons versus rats that are just in a standard cage. So the rats that are at rat summer camp have more and better connections than the rats just in the regular cage. So really the non-synaptic transmission of information is important in neuroplasticity, and synaptic transmission of information. So there's a lot going on with learning.